Hello children and welcome to the world of science. This is chapter number 16, Heredity and Variation of Standard 9th, Science 2, lecture number 2. In this lecture we are going to cover up the structure of DNA and the structure of RNA, their similarities and differences. Myself, Kavita Coburne of Gokula Education Society English Medium School is going to present this lecture to you all. Now we will start with the chromosome first, where the chromosomes exactly are. In the previous lecture we have learned about the chromosomes. In the diagram you can see here the structure of cell is shown to you. Okay, in this cell we have the nucleus, and in this nucleus we have the chromatin fibers, that is a thread like structures it is. And when this cell is just undergoing a division, means it is get divided into two parts or it is forming two new daughter cells, that time this chromatin fibers, they are just thread like structures, they started becoming coil like structures. So these are here, it is coiled fibers. These type of chromatin fibers are formed, and these fibers are just engaged or they are engulfed, they are just compiled up in the chromosomes. So like this chromosome structure we have studied in the previous lecture. This chromosome contains these chromatin fibers, and different structures of chromosomes you have learned, different shapes of chromosome you have learned. Now in this chromosome you have learned about that they are consist of two arms, P arm and Q arms. These are the two arms of it. It consists of chromatin fiber. This chromatin fiber is nothing but DNA it is. Okay, now the structure of the chromosome is how it is dumbbell shaped. Dumbbells, you know that. Okay, so it is of dumbbell shape, and in human beings you have learned there are total 46 chromosomes. They are paired one, so you will get 23 pairs of chromosomes in your cell. Now out of these 23 pairs we have learned we have two different types. One pair is totally different and the 22 pairs are same it is. So these 22 pairs are called as a autosomes, and the one single pair which is different, they are called as a sex chromosomes. Like these two types of pairs are there in our body, that is total 23 pairs in our pairs of chromosomes are present in the cell. Now one pair which consists of two chromosomes here, and this is 22 pairs, it consists of total 44 chromosomes. So 44 plus 2 is 46 chromosomes are present in the cell, and 23 pairs are there. This it is different in male and female, and this rest one autosomes are near about same in male and female. This much we should know for the further study. Now we will just continue with the next one, that is the DNA. As I told, the DNA is present in your chromatin fibers, that is in the ribosomes. So DNA's rough structure, just an introduction it is there. Now here you can see that is the rough structure of your DNA. It look like this. Okay, now it is the full form of DNA is deoxyribonucleic acid. Okay, and it was just found out by the Swiss biochemist, that is his name was Friedrich Mischer. He found it in 1869. Actually he was doing the study of white blood cells, and then he come to know about that DNA is also present in the nucleus of cell. So this he has given as a nucleic acid. DNA is also called as a nucleic acid. Then it was later found out, when he found out that it is in nucleus, but after some studies it was found out that this nucleic acid or DNA is present in the other parts of cell too. Now in the structure you can see here the lines are there. Okay, these lines, they are consisting of the genes, that is they are the segments, it consists of gene, and gene is the basic unit of this DNA. So this gene or the DNA is called the structural and functional unit of your body. So why the structural? Because it is giving the shape and size to your body, and it is just performing all the functions of your body, that is the growth and reproduction. So that's why genes or DNA is called as a structural and functional unit of your cell. What these genes are doing, these genes are just transforming, or information about protein synthesis is stored in genes. Protein synthesis you will learn it in the next coming chapters or in the coming topics over there. Okay, that protein is the basic or the building block of your body. So for the formation of proteins, this information is stored in this genes. Genes are present in these segments. Later in the next we will learn all these things. What these genes are also doing, they are just transferring the hereditary characters from parents to the offsprings. Means your color, skin color, hair structure, shape of your eyes, lips, face, physical structure, like these characteristics are there. Okay, and these are just carried out from parents to offspring through these genes, which is present here in these segments, and which these segments are present in this DNA. DNA is present in chromosome, chromosomes are present in nucleus, and nucleus is present in cell. Like this, these genes are very important, and so much protection is given to these genes in the DNA. Okay, now we will learn the structure of DNA. Here is the structure of DNA we have to study. All the things is written together in front of you, but we have to go step by step and you have to observe it clearly. Here the structure, if you will see, it is a double stranded structure. Just now we have saw the DNA, like this structure it is there, and all the details are written over here. We will see all one by one. First of all, this DNA molecules, they are called as a master molecules. Why they are called as a master molecules? Because its function is to control the functioning of your body, to bring out the growth in your body, and also it has to bring out the reproduction or the division of cells. All these things are performed by this DNA, and that is only responsible for it. That's why it is called as a master molecule. Okay, now one by one we will see in details about the DNA. Here in front of you, the structure which is given to you, which is shown to you, this structure is given to us by the scientists, that is called, their names were Watson and Crick, and it was proposed, the structure of the DNA, by them in 1953. So they told that this type of structure we are having of DNA molecule, that is a model, we can say. As per the model, you can see here two parallel strands are there. I have written here S1 and S2. These two parallel strands, they are coiled up. If you take the ladder, okay, and if the ladder is flexible one, if you just little bit twist it, then you will get this type of DNA structure in front of you. So here it is a two parallel strands which are coiled up, and this DNA structure is formed. So its name they have given as a double helix. Here this one full structure, it is double helix. These two strands are there, S1 and S2. I have written here strand 1, strand 2. They are twisted up like this. So like this it is made up of it. Okay, now here if you will just see one coiled with it, the length of this one is given here. Okay, this structure is given in your textbook also. Here also you will study. Now if you will just see here the one twist here. Okay, so one twist, its length is near about 3.4 nanometer. Okay, here from this turn to this one, here one twist we can say. So it is made up, its length is near about 3.4 nanometer. Now in this twist you will find in one twist near about 10 turns of nucleotides are there. These are the segments, they are called as the nucleotides actually. So here you will find total 10 turns in one twist. This distance between the two turns, we can say, two segments actually, here this one and this one, or any of the two you can take it. The distance between the two segments is near about 0.34 nanometer. This is what the structure is there, the length and breadth it is there. And here the width means from this end to this end, it is near about 2 nanometer. This much is the length and breadth of this DNA structure. The length total it is not given, it is just given the twist, just one twist. It is of how much length? 3.4 nanometer. The breadth here, it is from this end to this end, it is of 2 nanometer, and the length between the two segments is 0.34 nanometer. This is what the structure it is. Okay, now next what we have to learn is that what it is made up of. This total DNA, they are made up of some substances, and that substance what they are, that is deoxyribonucleotides. Okay, and these nucleotides are made up of deoxyribose sugar, nitrogenous base and phosphate groups. Actually, the two strands here, this two parallel strands, they are made up of deoxyribose sugar and phosphate group. That S and P, you can see here, S means deoxyribose sugar and P means phosphate group. So the parallel strands, they are made up of these two substances, and in between that you will find out these are the nucleotides which contains the genes. These are made up of nitrogen bases. Here the nucleotide is there, and these nucleotides are made up of nitrogenous bases. Nitrogenous bases, they are of two types now. One is purines and the next one is the pyrimidines. Now purines, they are also of two types. In purines you will find it out, they are made up of A and G. A stands for adenine, G stands for guanine. And pyrimidines, they are made up of thiamine and cytosine. Thiamine means T, cytosine means C. So purines are Ag and pyrimidines are Tc, like this here. So you can see here the A and T I have written and G and C it is. They are the nitrogenous bases. They are bonded together with the hydrogen bond. Means A and T, there in between them the bond is hydrogen bond. A and T is having double bond, G and C is having triple bond. It is of higher level. That is just for introduction I told to you. So like this nitrogenous bases you will find it out over here. Now you can see here the pairing I have written. That A pairs with T, G pairs with C. So adenine pairs with thiamine and guanine pairs with cytosine. Here vice versa, means A and T, it can be just transferred, it can form T and A also. It can be transferred, means we can say the complementary it is. So like these structures you can find it out here. There the pairings is fixed. A will not compile with C and G will not compare with T. So AT, GC, like this it is. This is for DNA. So in between them they have the hydrogen bond. Like this pairing you will get it, and in this only the genes are present, and that is just determining the color, structure, shape, size of the coming offspring. Whosoever will be formed, new formation if it will take, that then genetic information is contained in this nucleotides. So this is what the structure of DNA. DNA, it is consist of nucleotides, as I told here. See, that is made up of nucleotides. Nucleotides consist of genetic information. Where this is total genes are present in DNA, DNA is present in chromosome, chromosomes are present in nucleus, nucleus is present in cell, and cell are present in body. So this gene is so much important that it is totally protected by all this, and that is only responsible for transfer of characteristics from one generation to other. Now we have to see about the structure of RNA. RNA's full form is ribose nucleic acid. R for ribose, N for nucleic, and acid for A for acid. Now this is also one of the important nucleic acid in your cell. This is a second important nucleic acid. DNA was the first important nucleic acid, and RNA is your second important nucleic acid. Now there is a difference between RNA and DNA. DNA is double stranded and RNA is single stranded. Means here you will just find out only one strand. This RNA, it is made up of three types. Here rRNA, mRNA and tRNA. rRNA means that is ribosomal RNA, mRNA messenger RNA, tRNA means transfer RNA. Like these three types of RNAs you will get it. There is no types in DNA. So that is a difference. Means DNA it is of single type, RNA is of three types. DNA it is made up of double strand, RNA it is made up of single strand. This difference you have to just keep it in your mind. Now we will see what it is made up of. There is a little bit of difference in RNA and DNA contained. They both are made up of nucleotides only, but little difference it is. Here see, the RNA, it is made up of, that is same ribose sugar, phosphate molecules and nitrogenous bases. As like in DNA also we have that ribose sugar, we have the phosphate molecules, we have the nitrogenous bases. The only difference is in pyrimides and purines. Purines and pyrimides. Purines are same. They are adenine and guanine, and only the difference in pyrimides. In pyrimides there was thiamine in DNA, but in RNA you will find out instead of thiamine you have the uracil there, and cytosine is same. So that is only the difference in RNA and DNA, that RNA consists of uracil and DNA it is consist of thiamine. Other all the things are same in both DNA and RNA. Now we will see actually how the RNA is formed. RNA is a part of DNA actually. Here a rough structure it is shown to you just for your understanding. Actually this process is not that simple and not like this also. For our understanding this was shown to you. Here the cell is shown, here the nucleus, inside the nucleus chromosomes, inside the DNA is there. Now that RNA is forming from this DNA only. Okay, so here the mRNA is there, that is a part of DNA, single strand it is, that is coming out of this DNA. Okay, that mRNA which is carrying the information of protein synthesis from DNA to ribosomes. In the cell you have learned the structure of cell, that there are some ribosomes in the cell. So here the information is from DNA to ribosome. This is the ribosome structure. Here that one is carried by this mRNA, and that transfer of information of protein synthesis from DNA to ribosome, that process is called as a transcription. Here it is written. Okay, so what it is, that is carrying the information of protein synthesis of mRNA. DNA is carried from here to ribosomes. That process is called as a transcription. Okay, this is done by mRNA. M is messenger RNA. Okay, now when it is brought to this ribosome, in this ribosome this tRNA, one more type of RNA was there, that is called as a tRNA. That tRNA, what it is doing, that tRNA is bringing out the amino acids. Okay, three pairs of that acids they are just bringing out and just giving it in the ribosomes. Okay, it is supplying it. Here mRNA is bringing that strand from DNA to here, and tRNA is giving the amino acids in this process. So this process of transferring of amino acids for the protein synthesis is called as a translation. tRNA, it is giving out the amino acid for protein synthesis which is going to take place in this ribosome. So that process is called as a translation. Okay, now from here mRNA, that DNA, this type of single strand it is coming up. This will give you the amino acids to this, and then this ribosome RNA, that is rRNA, here in this ribosome only it is present. They are just doing the protein synthesis and then they will form this type of RNA. Okay, and that will take place in the growth and duplication or division of cells it is there. So here this total process is taken up in the cell with the help of, means here we can say in short that formation of RNA is taking place with the help of mRNA, tRNA and rRNA. Messenger is bringing the information, tRNA is giving the amino acids, and all the things together this ribosome is just making that rRNA. Means in short we can say that ribosome is like a mother, and these are the father and the parents or the family members. They are supplying the material, and here the mother is just cooking the food and just giving the ready food here. So here the RNA is formed. Like this RNA is there, and that is little bit different from dRNA. So DNA is double stranded, RNA is single stranded. Here the functions of mRNA, it is carrying the information of protein synthesis from DNA to ribosome, and that process is called as transcription. tRNA it is carrying the amino acids for protein synthesis, and that process is called as a translation. And what rRNA was doing? rRNA is performing the function of protein synthesis, that is the formation of RNA. Like this, 3 types of RNAs are working together and forming that RNA. Thank you for watching the video. Stay safe, stay home. Hope you must have understood it. The topics explained in this video. Your assignment is to read the lesson and revise the topics. Hope we will be meeting soon in the next video with the next coming topics of this lesson. Till then, take care, goodbye, and like, share and subscribe.